previously we did the quality control of the raw reads, and now we will do it for the alignment level. So like Eija already mentioned a bit, there are several things that you can check from after the alignment, just to see how things went. So first of all, there's the saturation of the sequencing depth, meaning that if we would be able to sequence more, would we be able to detect more genes or splice junctions? Then we check out the read distribution between different genomic features. So are the reads mapping to exonic or intronic locations, and where there? We also check if the read coverage is uniform along the transcripts, and it often is not super uniform because, like I mentioned with the FastQC plots, the random primers in the library preparation process can cause some bias to the uniformity or the coverage, and well, poly A capture and poly T priming do so too. And well, different genomic regions have different mapabilities because they are differently or more or less unique. So for example, if there are these repeated sequences, they are a bit trickier to map to. Again, there are several quality control programs for aligned reads. Here we list 4 of them, and RSeQC is the one that we are using today and the one that is available in Chipster. These are some example plots from the RSeQC that we have available in the Chipster. So the first plot here is the coverage uniformity. So here you have sort of the the gene body, and this is the number of the reads in that area. So this is a pretty nice example that the coverage is pretty uniform. Maybe a little bit of lacking coverage here on the beginning of the read. So if we would have some sort of bias here, it would look maybe a little more like this. And you get to see these soon when you do the exercises. Next, we have the saturation of sequencing depth. So here on red, we have the known junctions. So, okay, yeah, well, this is the number of junctions that are found. And on the x-axis, we have the percentage of total reads. So this is the full number of reads used, and then we use just smaller part of them to see at what point we have already found all the junctions. So from the known junctions, we pretty much know them already here at 30% of the reads. So adding more reads to our data, so sequencing more, wouldn't help us find any more known junctions. But the novel junctions shown here in green, they are still raising towards the end. So that means that if we would sequence more, we could find maybe a bit more new junctions. And well, blue is just a summary of these two curves. The third pie chart here is showing all the junctions that we have in the data. So in blue we have the known junctions, and then on green we have the completely novel junctions, and red is partially novel. So completely novel means that both ends of the junctions are new one, and partially novel means that out of the 2 ends of the junction, one is known and one is a new one. In Chipster and in general, this RSeQC tool takes in the BAM file, and then it can take this bed file as an input. In Chipster, we already offer you some of the bed files. They are created from ENCODE genes, so you can use those.
Ensemble.
Oh, sorry. Yeah, what did I say? Ensemble bed files, and you can also bring your own bed to Chipster if you want to. You can obtain those from, for example, from the UCSC table browser. Just make sure that the chromosome naming is similar or the same in the BAM file and in the bed file. So there are sort of 2 naming types available, either just using the number or then CHR number for the chromosome. So just make sure that they are the same, and we have tools to switch it if it's not correct in the beginning. Okay, so then when you run the RSeQC tool, you get a result file from there that can be in the beginning a bit heavy looking and a bit confusing too, unfortunately. So it starts with all number or read counts, and when actually they are not read counts, but they are the alignments. So as you remember, Eija was explaining that you have this number of reads, but you can have more alignments because a read can align to multiple locations in the genome. So that's the first thing that is a bit misleading there. Then you have a bunch of numbers there. You have the total records, and then some like numbers that are for quality failed reads or possible duplicates. Then you have the non-primary hits, unmapped reads, and then those reads that are below a certain quality cutoff, and the default for that is 30. Then the file continues, and you have here total reads and total tags, and then numbers for how these reads and alignments are divided into different locations in the genome, so whether they are in the exon area or in the intron or so on. So like mentioned, it can be a bit confusing, so I made this helper picture here in below. So if we now, you can see that there are 3 different reads here, A, B, and C. But we have 7 total records, so looking at this number here. Because blue read is actually mapping into 4 different locations, and the red one is mapping into 2 different locations. So thus we have 4 non-primary hits, these 4 here, because you know they are the extra hits there. The number of reads, the total reads, looking at this number here, that's 3, because yeah, we have 3 different reads in the data. And then the last number is the total tags. So tags now mean the part of the reads. So here would be a junction. So that's why we have 8 tags, because we have 1, 2, 3, 4, 5. Oops. 1, 2, 3, 4, 5, 6, 7, and 8. So this counts as 2 tags. Okay, I hope this is not too confusing. But just remember, this is the alignments. So the total number of alignments. These are the non-primary ones. And this is the number of reads. So the reads, so the sort of similar number that you saw in the FastQC plot. And then this is the number of tags. So when they are split in the junctions, that's when you count them as 2 or more.